Welcome to Chapter 9, Microbial Growth. In this chapter, we cover how microbes grow, oxygen requirements for microbial growth, the effects of pH, temperature, other environmental conditions, and media used for bacterial growth. Introduction. We are all familiar with the slimy layer on a pond surface or that makes rocks slippery. These are examples of biofilms, microorganisms embedded in thin layers of matrix material. Biofilms were long considered random assemblages of cells and had little attention from researchers. Recently, progress in visualization and biochemical methods has revealed that biofilms are an organized ecosystem within which many cells, usually of different species of bacteria, fungi, and algae, interact through cell signaling and coordinated responses. The biofilm provides a protective environment in harsh conditions and aids colonization by microorganisms. Biofilms also have clinical importance. They form on medical devices, resist routine cleaning and sterilization, and cause hospital-acquired infections. Within the body, biofilms form on the teeth as plaque, in the lungs of patients with cystic fibrosis, and on the cardiac tissue of patients with endocarditis. The slime layer helps protect cells from host immune defenses and antibiotic treatments. Studying biofilms requires new approaches. Because of the cell's adhesion properties, many of the methods for culturing and counting cells that are explored in this chapter are not easily applied to biofilms. This is the beginning of a new era of challenges and rewarding insight into the ways that microorganisms grow and thrive in nature. Section 1. How microbes grow. Learning objectives. Define the generation time for growth based on binary fission. Identify and describe the activities of microorganisms undergoing typical phases of binary fission, simple cell division, and a growth curve. Explain several laboratory methods used to determine viable and total cell counts in populations undergoing exponential growth. Describe examples of cell division not involving binary fission, such as budding or fragmentation. Describe the formation and characteristics of biofilms. Identify health risks associated with biofilms and how they are addressed. Describe quorum sensing and its role in cell-to-cell communication and coordination of cellular activities. The bacterial cell cycle involves the formation of new cells through the replication of DNA and partitioning of cellular components into two daughter cells. In prokaryotes, reproduction is always asexual, although extensive genetic recombination in the form of horizontal gene transfer takes place and will be explored in a different chapter. Most bacteria have a single circular chromosome. However, some exceptions exist. For example, Borrelia burgdorferi, the causative agent of Lyme disease, has a linear chromosome. Also, Agrobacterium tends to have two linear chromosomes. Binary fission. The most common mechanism of cell replication in bacteria is a process called binary fission, which is depicted in this figure. Before dividing, the cell grows and increases its number of cellular components. Next, the replication of DNA starts at a location on the circular chromosome called the origin of replication, or ORI, where the chromosome is attached to the inner cell membrane. Replication continues in opposite directions along the chromosome until the terminus is reached. The center of the enlarged cell constricts until two daughter cells are formed, each offspring receiving a complete copy of the parental genome and a division of the cytoplasm, cytokinesis. The process of cytokinesis and cell division is directed by a protein called FTSZ. FTSZ assembles into a Z ring on the cytoplasmic membrane. The Z ring is anchored by FTSZ binding proteins and defines the division plane between the two daughter cells. Additional proteins required for cell division are added to the Z ring to form a structure called the divisome. The divisome activates to produce a peptidoglycan cell wall and build a septum that divides the two daughter cells. The daughter cells are separated by the division septum, where all of the cell's outer layers, the cell wall and outer membrane, if present, must be remodeled to complete division. For example, we know that specific enzymes break bonds between the monomers and peptidoglycans and allow addition of new subunits along the division septum. Clinical focus, Part 1. Jenny, a 24-year-old pregnant woman in her second trimester, visits a clinic with complaints of high fever, 38.9 degrees Celsius, which is 102 degrees Fahrenheit, fatigue, and muscle aches, typical flu-like signs and symptoms. Jenny exercises regularly and follows a nutritious diet with emphasis on organic foods, including raw milk that she purchases from a local farmer's market. All of her immunizations are up to date. However, the healthcare provider who sees Jenny is concerned and orders a blood sample to be sent for testing by the microbiology laboratory. Why is the healthcare provider concerned about Jenny's signs and symptoms? Generation time. In eukaryotic organisms, the generation time is the time between the same point of the life cycle and two successive generations. For example, the typical generation time for the human population is 25 years. This definition is not practical for bacteria, which may reproduce rapidly or remain dormant for thousands of years. In prokaryotes, bacteria and archaea, the generation time is also called the doubling time and is defined as the time it takes for the population to double through one round of binary fission. Bacterial doubling times vary enormously. Whereas Escherichia coli can double in as little as 20 minutes under optimal growth conditions in the laboratory, bacteria of the same species may need several days to double in especially harsh environments. Most pathogens grow rapidly, like E. coli, but there are exceptions. For example, Mycobacterium tuberculosis, the causative agent of tuberculosis, has a generation time of between 15 and 20 hours. On the other hand, Mycobacterium leprae, which causes Hansen's disease, leprosy, grows much more slowly with a doubling time of 14 days. Microconnections. Calculating number of cells. It is possible to predict the number of cells in a population when they divide by binary fission at a constant rate. As an example, consider what happens if a single cell divides every 30 minutes for 24 hours. The diagram in figure 9.4 shows the increase in cell numbers for the first 3 generations. The number of cells increases exponentially and can be expressed as 2 to the n, where n is the number of generations. If cells divide every 30 minutes, after 24 hours, 48 divisions would have taken place. If we apply the formula 2 to the n, where n equals 48, the single cell would give rise to 2 to the 48th, or 281,474,976,710,656 cells at 48 generations in 24 hours. When dealing with such huge numbers, it is more practical to use scientific notation. Therefore, we express the number of cells as 2.8 times 10 to the 14th cells. In our example, we used 1 cell as the initial number of cells. For any number of starting cells, the formula is adapted as follows. N subscript n is the number of cells at any generation n, so that is equal to the product of n naught, which is the initial number of cells, and 2 raised to the n, the number of generations. The growth curve. Microorganisms grown in closed culture, also known as a batch culture, in which no nutrients are added and most waste is not removed, follows a reproducible growth pattern referred to as the growth curve. An example of a batch culture in nature is a pond in which a small number of cells grow in a closed environment. The culture density is defined as the number of cells per unit volume. In a closed environment, the culture density is also a measure of the number of cells in the population. Infections of the body do not always follow the growth curve, but correlations can exist depending upon the site and type of infection. When the number of live cells is plotted against time, distinct phases can be observed in the curve. And that's this figure. So initially we have the lag phase. The beginning of the growth curve represents a small number of cells, referred to as an inoculum, that are added to a fresh culture medium, a nutritional broth that supports growth. The initial phase of the growth curve is called the lag phase, during which the cells are gearing up for the next phase of growth. The number of cells does not change during the lag phase. However, cells grow larger and are metabolically active, synthesizing proteins needed to grow within the medium. If any cells were damaged or shocked during the transfer to the new medium, repair takes place during the lag phase. The duration of the lag phase is determined by many factors, including the species and genetic makeup of the cells, the composition of the medium, and the size of the original inoculum. The log phase. In the logarithmic log growth phase, sometimes called exponential growth phase, the cells are actively dividing by binary fission and their number increases exponentially. For any given bacterial species, the generation time under specific growth conditions, nutrients, temperature, pH, and so forth, is genetically determined, and this generation time is called the intrinsic growth rate. During the log phase, the relationship between time and number of cells is not linear, but exponential. However, the growth curve is often plotted on a semi-logarithmic graph, as shown in this figure, which gives the appearance of a linear relationship. Cells in the log phase show constant growth rate and uniform metabolic activity. For this reason, cells in the log phase are preferentially used for industrial applications and research work. The log phase is also the stage where bacteria are the most susceptible to the action of disinfectants and common antibiotics that affect protein, DNA, and cell wall synthesis. Stationary phase. As the number of cells increases through the log phase, several factors contribute to a slowing of the growth rate. Waste products accumulate and nutrients are gradually used up. In addition, gradual depletion of oxygen begins to limit aerobic cell growth. This combination of unfavorable conditions slows and finally stalls population growth. The total number of live cells reaches a plateau referred to as the stationary phase. In this phase, the number of new cells created by cell division is now equivalent to the number of cells dying. Thus, the total population of living cells is relatively stagnant. The culture density in a stationary culture is constant. The culture's carrying capacity, or maximum culture density, depends on the type of microorganisms in the culture and the specific conditions of the culture. However, carrying capacity is constant for a given organism grown under the same conditions. During the stationary phase, cells switch to a survival mode of metabolism. As growth slows, so too does the synthesis of peptidoglycans, proteins, and nucleic acids. Thus, stationary cultures are less susceptible to antibiotics that disrupt these processes. In bacteria capable of producing endospores, many cells undergo sporulation during the stationary phase. Secondary metabolites, including antibiotics, are synthesized in the stationary phase. In certain pathogenic bacteria, the stationary phase is also associated with the expression of virulence factors, products that contribute to a microbe's ability to survive, reproduce, and cause disease in a host organism. For example, Quorum sensing in Staphylococcus aureus initiates the production of enzymes that can break down human tissues and cellular debris, clearing the way for the bacteria to spread to new tissue where nutrients are more plentiful. The death phase. As a culture medium accumulates toxic waste and nutrients are exhausted, cells die in greater and greater numbers. Soon the number of dying cells exceeds the number of dividing cells, leading to an exponential decrease in the number of cells. This is the aptly named death phase, sometimes called the decline phase. Many cells lyse and release nutrients into the medium, allowing surviving cells to maintain viability and form endospores. A few cells, the so-called persisters, are characterized by a slow metabolic rate. Persister cells are medically important because they are associated with certain chronic infections, such as tuberculosis, that do not respond to antibiotic treatment. Now, pictured here are some growth curves from my own research that just to demonstrate that growth curves in real life aren't necessarily pretty. These were cells that were limited in their available nitrogen, so they did not have unrestricted access to nutrients. Sustaining microbial growth. The growth patterns shown in this figure take place in a closed environment. Nutrients are not added, and waste and dead cells are not removed. In many cases, though, it is advantageous to maintain cells in the logarithmic phase of growth. One example is in industries that harvest microbial products. A chemostat, cartoon of one being right here, is used to maintain a continuous culture in which nutrients are supplied at a steady rate. A controlled amount of air is mixed in for aerobic processes. Bacterial suspension is removed at the same rate as nutrients flow in to maintain an optimal growth environment. Measurements of bacterial growth. Estimating the number of bacterial cells in a sample, known as a bacterial count, is a common task performed by microbiologists. The number of bacteria in a clinical sample serves as an indication of the extent of an infection. Quality control of drinking water, food, medication, and even cosmetics relies on estimates of bacterial counts to detect contamination and prevent the spread of disease. Two major approaches are used to measure cell number. The direct method involves counting cells, whereas the indirect methods depend on the measurement of cell presence or activity without actually counting individual cells. Both direct and indirect methods have advantages and disadvantages for specific applications. Direct cell count. Direct cell count refers to counting the cells in a liquid culture or colonies on a plate. It is a direct way of estimating how many organisms are present in a sample. Let's look first at a simple and fast method that requires only a specialized slide and a compound microscope. The simplest way to count bacteria is called the direct microscopic cell count, which involves transferring a known volume of a culture to a calibrated slide and counting the cells under a light microscope. The calibrated slide is called a Petrov-Hauser chamber, as pictured here, and is similar to a hemocytometer used to count red blood cells. The central area of the counting chamber is etched into squares of various sizes. A sample of the culture suspension is added to the chamber under a cover slip, and that is placed at a specific height from the surface of the grid. It is possible to estimate the concentration of cells in the original sample by counting individual cells in a number of squares and determining the volume of the sample observed. The area of the squares and the height at which the cover slip is positioned are specific for the chamber. The concentration must be corrected for dilution if the sample was diluted before enumeration. And for most bacterial cultures, if you don't dilute the sample first, particularly if we're talking about an alpha or gamma proteobacterium, for example, then you're going to have a really rough time because you're going to see way too many cells to be counted. So you most likely will have to dilute. Cells in several small squares must be counted and the average taken to obtain a reliable measurement. The advantages of the chamber are that the method is easy to use, relatively fast, and inexpensive. On the downside, the counting chamber does not work well with dilute cultures because there may not be enough cells to count. So what could you do to improve your results if this is the only tool you had? That's something that's not in the book that I will talk about in lecture, but overall, you could try to concentrate your cells, which imposes its own limitations, of course. Here's an example of fungal cells being viewed in a hemocytometer, fungal cells being much larger. These are eustolygomatous cells that are undergoing nitrogen starvation. That's why they have all those little vacuole-looking structures inside these long rod-shaped cells. Here's another version of it. Using a counting chamber does not necessarily yield an accurate count of the number of live cells because it is not always possible to distinguish between live cells, dead cells, and debris of the same size under the microscope. However, newly developed fluorescence staining techniques make it possible to distinguish viable and dead bacteria. These viability stains, or live stains, bind to nucleic acids, but the primary and secondary stains differ in their ability to cross the cytoplasmic membrane. The primary stain, which fluoresces green, can penetrate intact cytoplasmic membranes, staining both live and dead cells. The secondary stain, which fluoresces red, can stain a cell only if the cytoplasmic membrane is considerably damaged. Thus, live cells fluoresce green because they only absorb the green stain, whereas dead cells appear red because the red stain displaces the green stain on their nucleic acids. Another technique uses an electronic cell counting device, a Coulter Counter, to detect and count the changes in electrical resistance in a saline solution. A glass tube with a small opening is immersed in an electrolyte solution. A first electrode is suspended in the glass tube. A second electrode is located outside the tube. As cells are drawn through the small aperture in the glass tube, they briefly change the resistance measured between the two electrodes, and the change is recorded by an electronic sensor. Each resistance change represents a cell. The method is rapid and accurate within a range of concentrations. However, if the culture is too concentrated, more than one cell may pass through the aperture at any given time and skew the results. This method also does not differentiate between live and dead cells. Direct counts provide an estimate of the total number of cells in a sample. However, in many situations, it is important to know the number of live or viable cells. Counts of live cells are needed when assessing the extent of an infection, the effectiveness of antimicrobial compounds and medication, or contamination of food and water. So here's an example of an automated cell counter from Thermo Fisher Scientific. Okay, I don't have anything to do with Thermo Fisher Scientific, and this was not in the Openstax book, but it's a pretty cool device. It'd be really nice to have one. Plate count. The viable plate count, or simply plate count, is a count of viable or live cells. It is based on the principle that viable cells replicate and give rise to visible colonies when incubated under suitable conditions for the specimen. The results are usually expressed as colony-forming units per milliliter. That's CFUs per mil, CFU per mil, rather than cells per milliliter, because more than one cell may have landed on the same spot to give rise to a single colony. Furthermore, samples of bacteria that grow in clusters or chains are difficult to disperse, and a single colony may represent several cells. Some cells are described as viable but non-culturable and will not form colonies on solid media. For all these reasons, the viable plate count is considered a low estimate of the actual number of live cells. These limitations do not detract from the usefulness of the method, which provides estimates of live bacterial numbers. Microbiologists typically count plates with 30 to 300 colonies. Samples with too few colonies, less than 30, do not give statistically reliable numbers, and overcrowded plates, more than 300 colonies, make it difficult to accurately count individual colonies. Also, counts in this range minimize occurrences of more than one bacterial cell forming a single colony. Thus, the calculated CFU is closer to the true number of live bacteria in the population. There are two common approaches to inoculating plates for viable counts, the pour plate and the spread plate methods. Although the final inoculation procedure differs between the two methods, they both start with a serial dilution of the culture. And of course, you can always buy a machine to automatically count colonies and measure zones for you. Zones will come up in another chapter. Now there is a cell phone app that's a plate counter as well, and I've used that with a little bit of success. It isn't always about looking at the plate and physically counting colonies yourself. Serial dilution. The serial dilution of a culture is an important first step before proceeding to either the pour plate or a spread plate method. The goal of the serial dilution process is to obtain plates with CFUs in the range of 30 to 300, and the process usually involves several dilutions in multiples of 10 to simplify calculations. The number of serial dilutions is chosen according to a preliminary estimate of the culture density. This figure illustrates the serial dilution method. A fixed volume of the original culture, 1 ml, is added to and thoroughly mixed with the first dilution tube solution, which contains 9 ml of sterile broth. This step represents a dilution factor of 10, or 1 to 10, compared with the original culture. From this first dilution, the same volume, 1 ml, is withdrawn and mixed with a fresh tube of 9 ml of dilution solution. The dilution factor is now 1 to 100 compared with the original culture. This process continues until a series of dilutions is produced that will bracket the desired cell concentration for accurate counting. From each tube, a sample is plated on solid medium using either the pour plate method or the spread plate method. The plates are incubated until colonies appear. 2 to 3 plates are usually prepared from each dilution, and the number of colonies counted on each plate are averaged. In all cases, thorough mixing of samples with dilution medium, to ensure the cell distribution in the tube is random, is paramount to obtaining reliable results. The dilution factor is used to calculate the number of cells in the original cell culture. In our example, an average of 50 colonies was counted on the plates obtained from the 1 in 10,000 dilution. Because only 0.1 ml of suspension was pipetted onto the plate, the multiplier required to reconstitute the original concentration is 10 times 10,000. The number of CFUs per mil is equal to 50 times 100 times 10,000. The number of bacteria in the culture is estimated as 5 million cells per milliliter, or CFUs per mil. The colony count obtained from the 1 in 1,000 dilution was 389, well below the expected 500 for a tenfold difference in dilutions. This highlights the issue of inaccuracy when colony counts are greater than 300 and more than one bacterial cell grows into a single colony. So here is the pour plate method with my editorialized commentary on it. Essentially, you take your 100 microliter of your sample and you mix it with warm agar. And you pour the sample onto a sterile plate and then swirl to mix and then allow it to solidify. Plate incubated until bacterial colonies grow. I'm not a fan of this because it depends on how well your bacteria will survive the warm agar. Most will, and it also depends on being able to swirl it out to evenly distribute your mixture. This is the spread plate method, which I've done a lot more of, and where you take your 100 microliters, pour it onto the solid medium, and then use a plate spreader to spread it evenly around. And then you get bacterial colonies to grow 24 hours or later based on what the growth rate is for that organism. In the spread plate method of cell counting, the sample is poured onto solid agar and then spread using a sterile spreader. The process is repeated for each serial dilution preparation, and then you can get a count and estimate the number of cells in the original sample as mentioned before. A very dilute sample, drinking water for example, may not contain enough organisms to use either of the plate count methods described. In such cases, the original sample must be concentrated rather than diluted before plating. This can be accomplished using a modification of the plate count technique called the membrane filtration technique. Known volumes are vacuum filtered aseptically through a membrane with a pore size small enough to trap microorganisms. The membrane is transferred to a Petri plate containing an appropriate growth medium. Colonies are counted after incubation. Calculation of the cell density is made by dividing the cell count by the volume of filtered liquid. The most probable number, or MPN. The number of microorganisms in dilute samples is usually too low to be detected by the plate count methods described thus far. For these specimens, microbiologists routinely use the most probable number, MPN, method, a statistical procedure for estimating the number of viable microorganisms in a sample. Often used for water and food samples, the MPN method evaluates detectable growth by observing changes in turbidity or color due to a metabolic activity. A typical application of MPN method is the estimation of the number of coliforms in a sample of pond water or any kind of water. Coliforms are gram-negative rod bacteria that ferment lactose. The presence of coliforms in water is considered a sign of contamination by fecal matter, more particularly if they're fecal coliforms. For the method illustrated here, a series of 3 dilutions of the water sample is tested by inoculating 5 lactose broth tubes with 10 ml of sample, 5 lactose broth tubes with 1 ml of sample, and 5 lactose broth tubes with 100 microliters of sample, or 0.1 ml. The lactose broth tubes contain a pH indicator that changes color from red to yellow when the lactose is fermented. After inoculation and incubation, the tubes are examined for an indication of coliform growth by a color change in media from red to yellow. The first set of tubes, a 10 ml sample, showed growth in all the tubes. The second set of tubes, the 1 ml sample, showed growth in 2 tubes out of 5. In the third set of tubes, no growth is observed in any of the tubes in the 0.1 ml dilution. The numbers 5, 2, and 0 are compared with Figure B1 and Appendix B, which has been constructed using a probability model of the sampling procedure. From our reading of the table, we conclude that 49 is the most probable number of bacteria per 100 ml of pond water. Okay, don't drink that water, but that doesn't sound, like a cesspool or anything. Here is an example of the Coulter method, which uses the MPN to calculate not only coliforms, but it can differentiate fecal coliforms. The fecal coliforms will fluoresce in the large and small wells there, and while the coliforms, the total coliforms, just turn it yellow. Indirect cell counts. Besides direct methods of counting cells, other methods based on an indirect detection of cell density are commonly used to estimate and compare cell densities in a culture. The foremost approach is to measure the turbidity, the cloudiness, of a sample of bacteria in a liquid suspension. The laboratory instrument used to measure turbidity is called a spectrophotometer, pictured here. In a spectrophotometer, a light beam is transmitted through a bacterial suspension. The light passing through the suspension is measured by a detector, and the amount of light passing through the sample and reaching the detector is converted to either percent transmission or a logarithmic value called absorbance, optical density. As the numbers of bacteria in a suspension increase, the turbidity also increases and causes less light to reach the detector. The decrease in light passing through the sample and reaching the detector is associated with a decrease in percent transmission and increase in absorbance measured by the spectrophotometer. Measuring turbidity is a fast method to estimate cell density as long as there are enough cells in a sample to produce turbidity. It is possible to correlate turbidity readings to actual number of cells by performing a viable plate count of samples taken from culture having a range of absorbance values. Using these values, a calibration curve is generated by plotting turbidity as a function of cell density. Once the calibration curve has been produced, it can be used to estimate cell counts for all samples obtained or cultured under similar conditions with densities within the range of values used to construct the curve. Measuring dry weight of a culture sample is another indirect method of evaluating culture density without directly measuring cell counts. The cell suspension used for weighing must be concentrated by filtration or centrifugation, washed, and then dried before the measurements are taken. The degree of drying must be standardized to account for residual water content. This method is especially useful for filamentous microorganisms, which are difficult to enumerate by direct or viable plate count. As we have seen, methods to estimate viable cell numbers can be labor-intensive and take time because cells must be grown. Recently, indirect ways of measuring live cells have been developed that are both fast and easy to implement. These methods measure cell activity by following the production of metabolic products or disappearance of reactants. Adenosine triphosphate, ATP formation, biosynthesis of proteins and nucleic acids, and consumption of oxygen can all be monitored to estimate the number of cells. So here's an example from my own work of cultures that I used the spectrophotometer to estimate the density in that media. And then here is another tool, a spectrophotometer, that is excellent to have in a lab. Again, this is from Thermo Fisher Scientific, no affiliation, and this is not in the Openstax book, but the NanoDrop 2000 is great because it uses optical density ratios to give you an indication of concentration and purity of nucleic acids and proteins. Very convenient. Alternative patterns of cell division. Binary fission is the most common pattern of cell division in prokaryotes, but it is not the only one. Other mechanisms usually involve asymmetrical division, as in budding, or production of spores in aerial filaments. In some cyanobacteria, many nucleoids may accumulate in an enlarged round cell or along a filament, leading to the generation of many new cells at once. The new cells often split from the parent filament and float away in a process called fragmentation, which is pictured here. Fragmentation is commonly observed in the actinomycetes, a group of gram-positive aerobic bacteria commonly found in soil. Another curious example of cell division in prokaryotes, reminiscent of live birth in animals, is exhibited by the giant bacterium Epulopiscium. Several daughter cells grow fully in the parent cell, which eventually disintegrates, releasing the new cells to the environment. How does that like animal live birth? We don't disintegrate the mothers. Anyway, other species may form a long, narrow extension at one pole in a process called budding. The tip of the extension swells and forms a smaller cell, the bud that eventually detaches from the parent cell. Budding is most common in yeast, but it is also observed in prosthecate bacteria and some cyanobacteria. The soil bacteria Actinomyces grow in long filaments divided by septa, similar to the mycelia seen in fungi, resulting in long cells with multiple nucleoids. Environmental signals, probably related to low nutrient availability, lead to the formation of aerial filaments. Within these aerial filaments, elongated cells divide simultaneously. The new cells, which contain a single nucleoid, develop into spores that give rise to new colonies. Biofilms. In nature, microorganisms grow mainly in biofilms, complex and dynamic ecosystems that form on a variety of environmental surfaces, from industrial conduits and water treatment pipelines to rocks and riverbeds. Biofilms are not restricted to solid surface substrates, however. Almost any surface in a liquid environment containing some minimal nutrients will eventually develop a biofilm. Microbial mats that float on water, for example, are biofilms that contain large populations of photosynthetic microorganisms. Biofilms found in the human mouth may contain hundreds of bacterial species. Regardless of the environment where they occur, biofilms are not random collections of microorganisms. Rather, they are highly structured communities that provide a selective advantage to their constituent microorganisms. Biofilm structure. Observations using confocal microscopy have shown that environmental conditions influence the overall structure of biofilms. Filamentous biofilms called streamers form in rapidly flowing water, such as freshwater streams, eddies, and specially designed laboratory flow cells that replicate growth conditions in fast-moving fluids. These streamers are anchored to the substrate by a head, and the tail floats downstream in the current. In still or slow-moving water, biofilms mainly assume a mushroom-like shape. The structure of biofilms may also change with other environmental conditions, such as nutrient availability. Detailed observations of biofilms under confocal laser and scanning electron microscopes reveal clusters of microorganisms embedded in a matrix interspersed with open water channels. The extracellular matrix consists of an extracellular polymeric substance, EPS, secreted by the organisms in the biofilm. The extracellular matrix represents a large fraction of the biofilm, accounting for 50 to 90% of the total dry mass. The properties of the EPS vary according to the resident organisms and environmental conditions. EPS is a hydrated gel composed primarily of polysaccharides and containing other macromolecules such as proteins, nucleic acids, and lipids. It plays a key role in maintaining the integrity and function of the biofilm. Channels in the EPS allow movement of nutrients, waste, and gases throughout the biofilm. This keeps the cells hydrated, preventing desiccation. EPS also shelters organisms in the biofilm from predation by other microbes or cells, protozoans, white blood cells. Biofilm formation. Free-floating microbial cells that live in an aquatic environment are called planktonic cells. The formation of a biofilm essentially involves the attachment of planktonic cells to a substrate where they become sessile, attached to a surface. This occurs in stages as depicted here. The first stage involves the attachment of planktonic cells to a surface coated with a conditioning film of organic material. At this point, attachment to the substrate is reversible, but as cells express new phenotypes that facilitate the formation of EPS, they transition from a planktonic to a sessile lifestyle. The biofilm develops characteristic structures, including an extensive matrix and water channels. Appendages such as fimbriae, pili, and flagella interact with the EPS, and microscopy and genetic analysis suggest that such structures are required for the establishment of a mature biofilm. In the last stage of the biofilm life cycle, cells on the periphery of the biofilm revert to a planktonic lifestyle, sloughing off the mature biofilm to colonize new sites. This stage is referred to as dispersal. Within a biofilm, different species of microorganisms establish metabolic collaborations in which the waste product of one organism becomes the nutrient for another. For example, aerobic microorganisms consume oxygen, creating anaerobic regions that promote the growth of anaerobes. This occurs in many polymicrobial infections that involve both aerobic and anaerobic pathogens. The mechanism by which cells in a biofilm coordinate their activities in response to environmental stimuli is called quorum sensing. Quorum sensing, which can occur between cells of different species within a biofilm, enables microorganisms to detect their cell density through the release and binding of small, diffusible molecules called autoinducers. When the cell population reaches a critical threshold, a quorum, these autoinducers initiate a cascade of reactions that activates genes associated with cellular functions that are beneficial only when the population reaches a critical density. For example, in some pathogens, synthesis of virulence factors only begins when enough cells are present to overwhelm the immune defenses of the host. Although mostly studied in bacterial populations, Quorum sensing takes place between bacteria and eukaryotes, and between eukaryotic cells, such as the fungus Candida albicans, a common member of the human microbiota that can cause infections in immunocompromised individuals. The signaling molecules in quorum sensing belong to two major classes. Gram-negative bacteria communicate mainly using N-acylated homoserine lactones, whereas gram-positive bacteria mostly use small peptides. In all cases, the first step in quorum sensing consists of the binding of the autoinducer to its specific receptor only when a threshold concentration of signaling molecules is reached. Once binding the receptor has taken place, a cascade of signaling events leads to changes in gene expression. The result is the activation of biological responses linked to quorum sensing, notably an increase in the production of signaling molecules themselves, hence the term autoinducer. Biofilms in human health. The human body harbors many types of biofilms, some beneficial and some harmful. For example, the layers of normal microbiota lining the intestinal and respiratory mucosa play a role in warding off infections by pathogens. However, other biofilms in the body can have a detrimental effect on health. For example, the plaque that forms on teeth is a biofilm that can contribute to dental and periodontal disease. Biofilms can also form in wounds, sometimes causing serious infections that can spread. The bacterium Pseudomonas aeruginosa often colonizes biofilms in the airways of patients with cystic fibrosis, causing chronic and sometimes fatal infections of the lungs. Biofilms can also form on medical devices used in or on the body, causing infections in patients with indwelling catheters, artificial joints, or contact lenses. Pathogens embedded within biofilms exhibit a higher resistance to antibiotics than their free-floating counterparts. Several hypotheses have been proposed to explain why. Cells in the deep layers of a biofilm are metabolically inactive and may be less susceptible to the action of antibiotics that disrupt metabolic activities. The EPS may also slow the diffusion of antibiotics and antiseptics, preventing them from reaching cells in the deeper layers of the biofilm. Phenotypic changes may also contribute to the increased resistance exhibited by bacterial cells in biofilms. For example, the increased production of efflux pumps, membrane-embedded proteins that actively extrude antibiotics out of bacterial cells, have been shown to be an important mechanism of antibiotic resistance among biofilm-associated bacteria. Finally, biofilms provide an ideal environment for the exchange of extrachromosomal DNA, which often includes genes that confer antibiotic resistance. That brings us to the end of Section 1. Join me next time for Section 2, Oxygen Requirements for Microbial Growth. Okay, bye.